The overall goal of this procedure is to characterize the surface chemistry of nanoparticles using tunable resistive pulse sensing, or TRPS, and determine the Zeta potential of the material. This is demonstrated by characterizing DNA-modified nanoparticles.
This method can help answer key questions in the characterization of nanomaterials and the development of bioassays.
The main advantage of this technique is that you can complete particle-by-particle analysis. Each nanoparticle signal is translated into a Zeta potential measurement, providing a complete picture of the sample being analysed.
Though this method can provide insights into the characterization of materials, it can also be applied to other applications, such as the development of bioassays for the detection of cells, viruses, DNA and proteins.
Generally, individuals new to this method will struggle because it's not a simple inject the sample and wait for the answer. There is some input from the scientist needed.
To begin this procedure, vortex streptavidin-coated particles for 30 seconds. Before sonication for 2 minutes at 80 watts to ensure monodispersity. Dilute the streptavidin-coated particles 1 in 100 in PBST buffer to achieve a resulting concentration of 1 × 10⁹ particles per milliliter. Then, vortex the particles for 30 seconds. Based on a binding capacity of 4,352 picomoles per milligram, add the appropriate concentration of DNA to the particles for resulting concentrations of 10, 20, 30, 40, 47, 95, 140, and 200.